Good morning, students. Today, myself, Dr. Kamala, Assistant Professor, Department of Biochemistry and Nutrition. Today, our topic is green synthesis of silver nanoparticles and its application. Okay. Today, our workshop is going to be explain how to synthesize nanoparticles from using silver nitrate and how to analyze that silver nanoparticles using spectrophotometry and its application in dye degradation. Here, the major principle is plant extract contains so many phytochemical components which having an ability to reduce and stabilizing the nanoparticles. Now we are using neem leaf extract as a phytochemical source, which phytochemical may have an ability to donate electrons and causing silver ions to reduce into metallic silver and able to form silver nanoparticles. So materials required for this is plant material. Okay, then we have to extract the phytochemical constituent present in that, then use that material as a reducing agent to create nanoparticles by using silver nitrate. Okay, so now the procedure is first you have to synthesize the nanoparticles using neem extract. This process is denoted as green synthesis. Okay, so which phytochemical constituent have the ability to reduce the silver nitrate is maybe tannins, flavonoids, terpenoids, flavonoids, and phenolic components. So first you have to take the fresh leaves and just thoroughly wash with the distilled water and dry it for a few seconds. Then you have to boil it with double ionized water. Okay, after boiling of leaf extract, we have to use this solution as a reducing agent. Then we have to prepare different molar concentrations of silver nitrate. Here we are doing an experiment on 1 millimolar to 5 millimolar. Okay, 1 millimolar to 5 millimolar. 10 millimolar silver nitrate is not properly giving nanoparticles. Okay, so you have to take 1 millimolar, 2 millimolar, 3 millimolar, 4 millimolar, 5 millimolar silver nitrate, different concentration in different glass, and using magnetic stirrer and use the temperature 70 degrees centigrade. Okay, then after complete mixing of this solution, slowly drop by drop add the neem extract. Okay, so reducing property once optimized, the phytochemical components donate electrons to silver ions. Okay, then reduced silver ions to form silver atoms. Okay, then able to form nanoparticles. Once we got a brown color, visualized reddish brown color, then we stop the reaction. So within 30 minutes, you got a nice brown color solution. Then we analyze whether we are able to generate silver nanoparticles or not. We have to study the stability of the nanoparticles and analyze whether we are creating nanoparticles or not. So in our laboratory, we have a spectrophotometer. We use spectrophotometric analysis as the primary source to analyze the nanoparticles. UV spectrophotometry. Here the formed silver nanoparticles once color change into pale yellow to reddish brown color, we have to declare that silver nanoparticles are formed. How these nanoparticles are formed? Due to surface plasmon resonance, SPR. Okay, so this solution used as a test solution we have to characterize at absorption peak between 400 to 450 nanometers, then we are able to generate silver nanoparticles. Okay, this is once we have to check whether silver nanoparticles are prepared or not. Okay, we got a peak. In this way, we got a peak. Okay, so we can conclude that we are able to generate silver nanoparticles. The peak height, how much time we have to consume to prepare silver nanoparticle, the peak zone is reflecting proportionally to the time course, right? Within 30 minutes, we got a little bit of slope at 400 to 450. Okay, 2 hours time, 24 hours. 24 hours is the best time course to generate silver nanoparticles at maximum absorption 400 to 450 nanometers, right? One more point here, stability. The stability of this colloidal suspension is monitored through over a time. SPR peak position and intensity indicate nanoparticles are well dispersed and resistant to aggregates. See, if colloidal solution is properly performed, then it is stabilized for a prolonged interval. So within 24 hours, you got a narrow peak. Okay, those nanoparticles are retained in colloidal state in a prolonged period of time. But 30 minutes we got a peak, but these colloidal suspension cannot retain the nanoparticles as such for a prolonged time course. Okay, so this once you got a narrow sharp peak at 400 to 450 nanometers, okay, then these nanoparticles are good nanoparticles. They against resistant the aggregation. Okay, due to capping effect of the neem extract biomolecules. Okay, now we synthesize the nanoparticles. Further analysis happened with FTIR analysis and SEM analysis. FTIR analysis, okay, transformation, Fourier transform infrared spectroscopy. It is used for identifying the functional group responsible for reduction and stabilization of the nanoparticles. Okay, we got a broad peak at 3400 per centimeters. Then it is responsible for phenols and alcohols. Peaks are observed at 1600 and 1650 indicates carbonyl and carbon-carbon stretching. Okay. Peaks observed at 1000, 1200, suggesting CO stretching. See, basing on the above results, we get an interpretation, the shift in these peaks after the reaction suggesting that proteins and secondary metabolites in the neem extract are involved in capping of silver nanoparticles and preventing the aggregation. Okay, nanoparticle formation is resulted through UV and spectrophotometric analysis. Stability studies, we can go with the FTIR analysis, SEM analysis, scanning electron microscopy, provide information regarding morphology and size distribution of synthesized nanoparticles. These synthesized nanoparticles generally spherical polyhedral in shape. These particles are ranging 10 nanometers to 50 nanometers in diameter. Surface texture, thin layer of organic material surrounded by metallic core, capping by plant biomolecules. So in this way, we can analyze the nanoparticle distribution and synthesize by SEM analysis. Okay. Furtherly, we discussed about applications. In this lab, we just conduct dye degradation experiment. Okay. Nanoparticles effectively photocatalyst effectively act as a photocatalyst and degradation of organic pollutants like methylene blue and methylene orange. Okay, now the thing is, whatever nanoparticles are produced in the above strategies and analyze their stabilities. Furtherly, those nanoparticles are used to reduce the dyes as an impurities in water bodies. Silver nanoparticles transfers electrons to the donors and converted into acceptors, right? Photocatalysis under UV or visible light, silver nanoparticles undergo electronic excitation. The generated electron hole pairs react with water and oxygen to produce heavily reactive radicals. These radicals may have an ability to interact with the dyes and convert it into colorless intermediates. Furtherly, CO2 and H2O liberation happen. The high surface area to volume ratio of the nanoparticles increases the number of active sites and improve the decolorization of dye solution. This is one of the application explained in the lab. Apart from this, so many applications are executed with these nanoparticles. One is drug delivery system, wound healing, heavy metal removers in the water bodies, degradation of organic pollutants, nanofertilizers, nanopesticides, plant growth promoting molecules, catalytic applications, food packing applications. These are the applications for silver nanoparticles. This is a team along with me. They're working together and execute this class. Thank you very much. Thank you all.